back to It's Always Sunny in Raleigh. I'm your host, Hannah Covey. Today we're talking to an epigenetics specialist about recent controversies in her field. Welcome, Catalina. Let's start by having you tell our audience about epigenetics.
Sure. Well, the field of epigenetics has evolved throughout the years as new discoveries have been made. At the beginning, it was known as the molecular pathways that led to the expression of a genotype through a phenotype. Currently, the field is defined as a study of the changes in gene expression and its function, and how they are heritable from one generation to another, even though they do not change the DNA sequence.
That's really interesting work you're doing. Can you tell us a bit about epigenetics more in depth?
Sure. Let me start with the basics of genetics. Every cell in our body contains the same genetic material, the DNA, which if it's stretched out is approximately 6 feet long. Additionally, there are over 200 cell types in our body, but even though they contain the same DNA strand, the genes in each cell are different because one are expressed and other are repressed. For example, if we have muscle cells and neuron cells, they have the same DNA in their nuclei, but they are completely different in their function and appearance.
So you said that we have a vast amount of DNA in our bodies. How exactly is it stored in the nucleus of every cell?
Yes, if stretched out, the DNA is approximately 6 feet long. Therefore, it needs to be folded into a chromatin structure that allows it to fit into tiny cells. First, the DNA strand binds into a double helical structure. Then, the helix is wrapped around protein called a histone. Imagine this is a histone. The DNA binds around 8 of these histones, forming a nucleosome, and then it's sealed by another histone. The DNA will wrap around many histone octamers, forming a beaded string, which will coil up in different ways until it gets to the correct size to fit into these cells.
Okay, thank you. So what exactly controls our different gene functions?
Good question. So here's where epigenetics comes in. The expression of genes contained in our DNA is mediated by epigenetic marks that decide which genes are going to be expressed or repressed. Epigenetic marks include the addition of modifications to DNA, RNA, or proteins. DNA can be methylated as methyl groups bind to cytosine bases. Histones can also be methylated, acetylated, or phosphorylated. Also, respective groups can be added to their tails. MicroRNAs also affect the epigenetic marks and DNA and histones.
Okay, thank you for that explanation. Let us now welcome Anna, a nutritionist from Raleigh, to tell us a bit more about nutrition. Welcome, Anna.
Hi.
Can you tell us about how diet and epigenetics are related?
Sure. So the good news about diet is that it is easier to study because we understand how our bodies use the food that we put into them. The bad news is that diet is very influential during multiple developmental periods and can actually affect your offspring's genetic makeup.
Can you give us some examples of these developmental periods?
Sure. So pregnancy is where it all begins. Both the mother and the father contribute to the epigenome of the fetus. For the mother, a major contributor is the use of prenatal vitamins, which include folate, vitamin B12, and others. Without these vitamins, the risk of complications runs much higher. For example, folate deficiency is associated with neurological defects. Another contributor is undernutrition. Research has shown that when a woman is malnourished during pregnancy, she tends to have a child with a lower birth weight. The thrifty phenotype hypothesis says that low birth weight is correlated with a greater risk of health problems like diabetes and heart disease, because poor fetal nourishment leads to metabolic changes that increase their chances of survival. And this is good when the conditions stay poor, but not when nourishment has improved.
So does the mother contribute as much to her offspring's epigenome in other species?
A great example is the agouti gene in mice. Research has shown that the diet of a female mouse contributes to the condition of the agouti gene. A methylated agouti gene typically produces offspring with normal coat color and weight with a low risk of disease. On the other hand, an unmethylated agouti gene produces overweight offspring with abnormal coat color that are more likely to develop disease. Catalina, can you explain methylation to the audience?
Sure. As I briefly explained before, the addition of a methyl group to the cytosine basis of the DNA is called methylation. If we have a DNA strand with the following bases, methyl groups would be added next to the guanine bases. Here, here, and here. This junction of cytosines and guanines next to each other, it's called CPG islands. When a methyl group binds to a CPG island, they turn off genes because transcription cannot occur, given that it becomes an obstacle.
Thank you for that clarification. Now back to what Anna was saying. Are fathers as influential as mothers with respect to diet and the epigenome?
So the fathers do contribute, but not as significantly. One example of paternal influence is that the diet of young men can actually affect their grandchildren. A study actually showed that food scarcity was correlated with a greater lifespan for the grandchildren, whereas food abundance was correlated with a shorter lifespan. Another example is the insulin-like growth factor 2 gene, which the fetus receives the transcribed gene only from the father. And research has shown that overweight fathers have less DNA methylation at this gene, which would obviously affect what is passed on to the offspring.
So how do the effects of diet change when we focus more on later developmental periods?
Research has shown that epigenetic changes due to the mother's diet during the postnatal period are likely causes of increasing obesity. One problem is overnutrition, which is often the case with formula-fed children. They tend to show quicker growth than breastfed children during this time period, which is associated with a higher risk of obesity and cardiovascular problems.
So now that we've heard about earlier developmental periods, how does diet affect the epigenetics of teenagers and adults?
Even though folate is important to fetal development, it continues to be very important through adulthood. So dietary folate is found in green vegetables like broccoli and lettuce. It helps to maintain a healthy DNA methylation pattern, and it can also repair DNA damage that has accumulated. Another good practice is the use of antioxidants. Anthocyanin is an antioxidant that is known for its red and purple color. It's found in things like cherries, kidney beans, and other foods. So this works by removing the free radicals that actually damage the DNA. Another common antioxidant is EGCG, which is found in green tea, and it regulates the DNA methylation of specific genes.
One topic our adult viewers would be interested in is the use of alcohol. How does alcohol affect epigenetics?
There has been no evidence to suggest that casual alcohol use affects the epigenome. However, chronic alcohol use has been shown to cause health problems like folate deficiencies, vitamin B deficiencies, DNA hypomethylation, and other serious health problems.
Okay. I had no idea diet was so complex. Thank you, Anna, for telling us about this. Let us welcome Caitlin, a physical therapist from Raleigh, to discuss the role of exercise. Welcome, Caitlin.
Hi, thank you for having me. The study of how exercise affects epigenetics is just beginning. Exercise affects epigenetics primarily through alteration in DNA methylation. These alterations determine whether or not a specific gene is turned on.
We've talked a lot about methylation on the show today. Can you explain where methylation occurs for our viewers?
Yes, exercise specifically targets DNA methylation in the brain, blood, skeletal muscle, cardiac muscle, adipose tissue, and buccal cells. All of these areas show changes in methylation patterns. However, there is limited evidence on the long-term effect exercise has on these areas. Some studies suggest that these methylation patterns affect insulin production and LDL levels throughout the body.
So how much exercise is necessary to see these epigenetic results?
After merely 6 months of aerobic exercise, the entire skeletal muscle genome undergoes significant changes in methylation. The adipose tissue also shows great variation in DNA methylation from before the exercise took place, which again could correlate to a decrease in insulin levels. The 6 months of aerobic exercise also directly affects lipogenesis, which is the metabolic breakdown of fats.
Do you have to participate in long-term exercise programs in order for it to have an impact in your epigenome?
Actually, you do not. A study recently found that both short and long-term exercise patterns lower DNA methylation. For example, exercising for 3 hours will directly affect the genome's methylation patterns, protecting the body from disease. The single bout of exercise can remove the methyl groups on genes involved in glucose transport and mitochondrial regulation, allowing transcription to occur. These exercises signal the demethylation of regions involved in metabolism, such as enhancers, which are DNA-based pairs that increase the likelihood of transcription occurring.
Does exercise affect DNA methylation in areas not directly involved in metabolism?
Yes, a study conducted showed the immediate effect of exercise throughout the brain, affecting significantly the hippocampus, which I have the skeleton right here, and Catalina can help me out with this. The hippocampus is located on the back portion right through here of the skull, and then the cerebellum is more central, but on the inside right around through here of the skull, and both of these areas are affected by the DNA methylation. This led to an increase in brain neurogrowth factor in the hippocampus. I have actually put together a video to show you all how a human study was conducted to specifically target the effects of exercise on the epigenome.
So I'm here to mimic a study that was conducted where only one leg pedals the bike.
And this was done for 45 minutes for 3 months. And we only did one leg so that you could tell about the different methylation patterns, and this wasn't affected by nutrition or anything because only one leg was tested. And after this was done, there were over 5,000 new methylation patterns in just this one leg compared to this other one. So the main regions that were affected were enhancer regions, which they alter the insulin levels, the inflammation, and the metabolic areas. And so these DNA methylation changes focused on these few areas the most after the study was conducted.
I think it's safe to conclude that physical exercise is capable of changing DNA methylation and gene expression throughout the epigenome. We've heard a lot today about epigenetics and its effects, but there's been a lot of controversy as well. Recently, an article was published by Duke University suggesting that epigenetics has linked poverty with conditions such as depression, anxiety, and drug dependency. However, many people say that these studies are often hard to control for confounding factors and are not accurate. Let's now take some calls from our viewers about questions from today's show.
I'm a person of low income and also have diabetes. How can this affect my children and future generations?
So this is something that we have all been studying, and current research has shown that diseases and traits can be associated with your socioeconomic class. Having less access to good nutrition, as well as education about its importance, can lead to increased prevalence of diabetes in these areas due to the insulin factor we mentioned earlier. Try to find low prices at your local farmers market and try to exercise more to limit the epigenetic effects in later generations.
Let's take another call.
I was just wondering if the environment influences epigenetics.
Environmental toxins have been shown to have epigenetic effects. For example, cigarette smoke in the air. High levels of smoke can result in genetic mutations, leading to higher risks of cancer in the following generations. Generations down the line, it will be very difficult to identify who was the responsible and what source caused the issue. Luckily, our government has put laws in place like the Clean Air Act that regulate chemicals, ensuring that they can be taken care of properly.
Let's take another call.
I recently had some genetic testing done and the results were somewhat unsettling. I do not want this information being passed along to my boss, but am unsure of what I am obligated to reveal. Do you have any advice?
Hi, in my physical therapy practice, we pride ourselves on being discreet with our patients' health information. It is a huge challenge to protect medical information today, and after being tested for epigenetic changes, you may want to prevent this information from being told to an employer or a friend, depending on the results. My advice would be to talk it over with your doctor to understand what will happen in the future, but keep it disclosed from your employer.
And let's take one more call.
I have recently found out I have a genetic condition. Am I obligated to tell my boyfriend who I may have children with in the future?
So that is a very interesting question. Legally, you're not required to reveal any health information that you don't want to. However, you might feel morally inclined to notify others who could get that disease or trait, especially when children may be involved.
Thank you for all the calls that we've gotten today. I want to thank everyone for joining me today on It's Always Sunny in Raleigh. Please tune in next week and have a great night.